Hi there, Professor Stafaro, and today's screencast is on oxidative phosphorylation. This is the last part of our cellular respiration pathway, and here we're going to describe the major events of the electron transport chain as well as describing chemiosmosis. So here's our diagram of cellular respiration, and we are in the third and final step. This is called oxidative phosphorylation. It occurs in the mitochondria and specifically in the inner mitochondrial membrane. You can see that oxidative phosphorylation consists of both the electron transport chain and chemiosmosis. And the other thing that we can see here is that we get a bigger ATP bubble at the end. And that means this is where we're going to produce the bulk of the energy in this process. So let's go back and look at a mitochondria. Mitochondria have two internal compartments that are created by the inner mitochondrial membrane. So we have the inner membrane space, that's the space between the outer membrane and the inner membrane. Then we have the mitochondrial matrix. This is the center of the mitochondria, the larger space. This is where the Krebs cycle took place in the mitochondria. It also contains DNA and other enzymes that are used in cellular respiration. And then we have the folds of the inner mitochondrial membrane themselves. And it's this inner membrane that we want to focus in on. So let's use the little magnifier to focus in on that inner membrane, and it should look like this if we were magnifying it. And we should see that the inner membrane, there are a series of proteins that are distributed here. Those are the purple blobs. And these proteins, they're called cytochromes, are going to be used to shuttle or transport electrons that are coming off of hydrogens, and that essentially is the electron transport piece. And as a result of shuttling or moving these electrons, we're going to get 90% of the ATP that's produced by cellular respiration. So let's take a look at this figure a little bit more and name the parts. So first, find the inner membrane. This is where those cytochromes are located. They are going to be the transport protein. Then find NADH and FADH2. They are in the position to give off the hydrogens. And remember, hydrogens have a proton and an electron. Protons are positive, electrons are going to be the negative part of the hydrogen. If we follow the orange arrow, this follows the path of the electrons as they shuttle or transport through those cytochrome proteins. The hydrogen protons, the positives, are shuttled into the inner membrane space. And this is going to create a potential energy gradient that's going to be important in producing ATP. Last, on the far right, is what's called the ATP synthase. It's an enzyme in that inner mitochondrial membrane, and it is the passageway for the hydrogen protons to cross the membrane. Remember, things have to move from a high to a low concentration, and the only way the hydrogens are going to cross is through that ATP synthase. So we utilize that energy to form ATP. One more important piece is oxygen. Oxygen is going to serve as the final electron acceptor. It essentially clears the deck. As the electrons move through those purple blobs, we're following that orange arrow, you can see that it ends up on oxygen. And oxygen clears the way for other electrons to enter into the electron transport chain so that more ATP can be made. If we use the animation here, we can see that the electrons, the negatives, get moved from one transporter to the next, and the protons, the positive parts, get put into the inner membrane space. Essentially, the positive and the negative part of the hydrogen is being separated in this process. All right, so now we have this hydrogen gradient that's in the inner membrane space, and the process of moving it from a high to a low concentration is referred to as chemiosmosis. Chemiosmosis drives the protons through the ATP synthase, and this enzyme then produces ATP. So let's take a count of how many ATP get formed at each part of cellular respiration. At glycolysis, we had a net 2 ATP form, but we also produce NADH, and that NADH ultimately went to the electron transport chain. For every 1 NADH, you're going to get 3 ATP formed. So 2 NADH produced at glycolysis leads us to 6 ATP that can be formed in the electron transport chain. Acetyl-CoA, when it is formed from pyruvate, 2 NAD are produced, and again we get 6 ATP formed at the electron transport chain. In the citric acid cycle, we get NAD and FADH2 produced. So that in oxidative phosphorylation, those 6 NADH that are produced lead to 18 ATP, the 2 FADH2 lead to 4 ATP, and then we get 2 ATP formed as a part of the citric acid cycle process. Now the reason why you see the 2 next to acetyl-CoA and the citric acid is that this is a doubled process. Remember, we ended up with 2 pyruvate, and so we have a doubled process going into acetyl-CoA formation, citric acid cycle. So let's look at it in a picture form. Again, we see the 2 ATP that are formed and the 2 NADH. Then at acetyl-CoA, we see the 2 NADH formed. At citric acid, we see the 6 and the 2 FADH. And all of those electron carrier molecules are used by the electron transport chain to generate that huge amount of ATP. So that's where we get our 38 ATP per 1 glucose molecule. Let's take a look at the effect of poisons on the electron transport chain. There are 3 different categories of poisons to cellular respiration, and we're going to focus in on those associated with the electron transport chain. So the first one is rotenone. Rotenone is an insecticide. It binds to an electron carrier at the beginning of the electron transport chain and prevents electrons from moving through the chain. Cyanide does a similar thing, only it blocks the last electron carrier protein from dumping that electron onto oxygen. Then we have ogleomycin. Ogleomycin is an antibiotic, and it's used to fight fungal infections. And here, ogleomycin will block the ATP synthase, so hydrogen protons, we can't utilize that potential energy created in that gradient. They won't be able to move through that synthase, and ATP won't be produced. And then lastly is the DNP. This stands for dinitrophenol, and it abolishes the hydrogen ion gradient by making the inner membrane leaky to hydrogen protons. So you abolish the potential energy gradient that's going to be used to make ATP in the first place. All right, that summarizes electron transport. Let's watch the animation to summarize the process one more time, and I'll see you in class. Ta-ta!
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.